First off, you start off with a little zygote, which contains how many cells? One cell. And then you do a couple of stages or rounds of cleavage. And in a cleavage event, basically what we're seeing is we're seeing the cells divide. So you're going through multiple cases of the cell cycle. In fact, you go through 3 cases of the cell cycle in order to form 8 cells. So this stage right here to me would be an 8-cell stage. And you probably could see that from the fact that there's 4 on this face, so there's got to be 4 on the opposing face. Now you would go through more rounds of cleavage, which basically means more rounds of the cell cycle, more mitotic events basically, and end up with a variety of cells that actually is a very, very hollow ball of cells. This is called a blastula. This right here is what I would call a cross-section of a blastula. Make sense? Now this event that we're seeing right here, and this is kind of the event that's happening, you can see where some of those cells are kind of being pushed inward, and that event that we're seeing right here is called gastrulation. The infolding of these cells to form our different tissue layers. In the end, we're left with a gastrula. And we can identify some different points, like for instance, we've got on the very, very outside, ectoderm. On the very inside, right about in there, this would be endoderm. Endoderm right there. This space that's empty right through here is called the blastocell. And we could trace that to existing right here. It's kind of like the open space between your cell layers. And we could even trace it to this area that is right through there. So this area that I'm pointing to there is still going to be the blastocell. Bless you. So, yeah. Cool. And of course, the step before the formation of the zygote would be the sperm and the egg coming together. Fusion, right, of your 2 haploid cells. So I think we probably understand how that happens. Right here, these 2 guys that you're seeing there represent your different tissue layers. So you form the different tissue layers at that point. Cool. Okay. So there's that. I think we're all done with that picture. Let's talk about a larval stage, which is an event in which you have, you have a small minimized form of that creature that is going through metamorphosis to go into the adult form. So this would be going from the larva, which is the sexually immature stage, I don't know why I wrote equals there, that doesn't really make much sense, but to the adult phase. Maybe instead of writing larval stage, I don't know why I was thinking that. Maybe we should just write metamorphosis, because that's probably like better describing this event. Going from the larval stage, the sexually mature stage, to an adult phase, that would be metamorphosis. Some creatures have more than one, some creatures just have one. It kind of depends on the animal. One metamorphosis, going from like an immature to an adult stage. Let's see. Animals is a group evolved from the choanoflagellates, which are a type of protist. So our ancestors are traced back to the Protist kingdom, or what used to be the Protist kingdom. We had mentioned this the other day, but I'll kind of mention it again. The first animals evolved, and this is fairly approximate, but between 700 and 850 million years ago. So animals have been around for a little bit less than a billion years. Of course, the first animals that did evolve were not what we would consider animals. They're sponges. What phylum is that? There you go. So there's your first phylum. That would be from the animal kingdom. I think we want to remember what's happening in the Cambrian explosion. Not really like an explosion, like as in what we would classify an explosion or an explosion is. It's a time of great diversity. Like a what? I guess. About 530 or so million years ago. And the reason that it happened was because of predator and prey relationships. What's that? Diversity bomb? I don't think it's a diversity bomb. Can't wait to listen to it on YouTube, Joseph. An increase in O2 in the atmosphere. And the Hox genes increase in complexity. Right? Your first land animals were about 460 million years ago. The first vertebrate land animals was about 400 million years ago, maybe 350 million years ago. Yeah, it makes more better sense. It's about 100 million years difference. Cool. It's kind of just like your general, like, massive events of animal evolution. Okay. Ow. You're joking. Okay. So let's talk a little bit about symmetry. And maybe to help us with this process, I've got a fun-filled picture that would assist us. Hopefully you like it. I don't think everyone would like it though, but I think I like it. So I'm going to give you a couple things to copy it down. So yeah, let's make this happen, and let's say we'll start back up in 35 seconds. Okay, sorry, Connor. Let's keep this going. I see two types of symmetry here. In the first case, I would say that we have radial symmetry. So let's draw that out. Let's write radial right here. And in the second case, I would say that we have bilateral symmetry.
Are you going to explain the differences?
In radial symmetry, you can have many cuts, but still many cuts, but there's still symmetry. Think of like a pizza or this, whatever that is, pot, or this little creature, looks like a cnidarian. You could cut it any different way you want to, and you're going to have symmetry on either which side. And that's as opposed to bilateral symmetry, in which case there is only one single line of symmetry. Kind of our difference there. Now, if a creature does have bilateral symmetry, then it's therefore supposed to have a couple of other features that we need to mention. One is that there's going to be a dorsal top and a ventral bottom. So there's a top side and there's a bottom side. Oh. Of course, there's a right and left. I'm not going to write that down. There would be an anterior, which refers to the head. And there's a posterior, which would be more tail. You have to think of an organism that's like on all fours. Oh, please stop. Thank you. Okay, anterior head and posterior tail. And dorsal top, ventral bottom. Okay. And let's use the term cephalization. What does that mean? Okay, that's when you have tons of nervous tissue. At the head of the creature. The more cephalization, then the more advanced the creature is as far as like brain-wise. I guess that's the best way to say it. Okay, we have another picture to draw. I'm sorry there's so many pictures today, but that's okay, I think. So here's the next picture. I'll zoom in a little bit. Let's say you have 41.5 seconds to draw this out. Okay, so let's take this picture and let's maybe just talk about it a little bit. But maybe off to the side, we might want to do just a little bit of vocab real quick to go over some tissue layers. So I'm going to get you to write down the terms ectoderm, endoderm, and yeah, let's say mesoderm. And then let's jot down the terms, maybe over to the side as well, diploblastic and triploblastic. Okay. So ectoderm, I think, would be the outside tissue layer. Covers the surface. The endoderm would be the innermost layer. And mesoderm, of course, would be kind of the middle layer of tissue. It may or may not be present. As diploblastic creatures, if they're diploblastic, that means they have two tissue layers. That's what the di there means, two tissue layers. They would have both endo plus ectoderm. And if you're triploblastic, then you would have endo plus ecto, as well as the middle mesoderm. Now, as you could guess, diploblastic creatures would be like pretty old or pretty old school style, and triploblastic would be like a newer approach. So, like your lesser evolved creatures would be diploblastic, and the more evolved creatures would be triploblastic. I don't know why I drew that arrow there, is that that's kind of the trend, is to become more triploblastic. And then I want to talk about what a coelom is. So this is another term that I want us to write down, a coelom, which is a body cavity that is lined with mesoderm. Coelom, C-O-E-L-O-M. And if an organism contains a coelom, then that organism is called a coelomate. So the coelomate is the organism that has a coelom. And our three creatures that are over here go kind of in an order. We've got up top a planarian, which is going to be an acoelomate, which basically means that it's going to be not having a coelom. That's why the A is in front, no coelom at all. Okay, right there. And then this right here is going to be a pseudocoelomate. And then down here we have a perfect coelomate. And again, it's whether or not you have a true body cavity or not. Now, to kind of throw in some of your phylums, just because I think we might want to, this guy right here is a Platyhelminthes. Remember that stands for your flatworms, so there's a Platyhelminthes. That right there is a nematode. This is Nematoda, that's the phylum there, because it's a perfectly round worm. And then this would be what type of phylum? Yeah, because we see the segmented parts, so annelids right there. Now, though it's a little difficult to see, I want to kind of go over some like parts to this real quick. Right here in the middle, you have your endoderm. Out on the outside, you have your ectoderm. And that means that there's this kind of, the endoderm is that light color in the middle. It runs in a circle. The ectoderm is on the outside, that light color. That middle layer that kind of goes, that does maybe this movement, let me outline it in yellow. Tissue layer that's doing this little number right there. I don't know if that works out really well. Y'all see that? At least maybe moderately well. That would be your mesoderm. And do you see, and it's a little hard to see, your book would have a better picture, of course, but do you see how there's this cavity that's formed right there? That cavity, that right there is your coelom. It's completely lined with mesoderm. And that's kind of where that definition's going, is completely lined with mesoderm. Now look at your pseudocoelomate up top, or right above this. You can see that cavity right here, this white space. So there's your coelom. And you'll notice that with my yellow marker, I've got mesoderm on the outside, but what's to the inside of that coelom? What tissue layer is that? That's the endoderm that's right there. So this cavity is not completely lined with mesoderm, therefore that's why it's a pseudocoelomate, a partially coelomate. Now, do you see a body cavity in the top one at all? I don't see a body cavity at all. I do see my, right here, I do see my ecto, my meso, and my endo, but I don't ever see a body cavity, a hole that's formed. Cool? Now, which do you think we are? You trace it. I would put us right here, correct? On the coelomate line? Okay. Well, that would make sense because when you open up our body cavity, is there just like stuff everywhere? No, I mean like you open up the body cavity and like there's just going to be blank space. Of course there's organs inside, but there is just blank space too as well, correct? Yes? Is there not some like pockets of air or whatever it might be that is between my liver and my kidney? Of course there is. So I'm a coelomate. Cool. That means I'm also having to be triple blastic if you think about it. Because if you're a pseudocoelomate or a coelomate, you for sure have to be triple blastic. And yeah, that's the way that works out. So I think that you probably want to be looking at this in your chapter a little bit more. we have the most simple amount of material to go over. I'll probably finish that during fourth period today. So make sure you watch this video this evening.